So now we'll start with today's topic, that's the cell signaling lecture 2. So this lecture is also very important. So in the previous lecture we have done the few important topics regarding the what are the GPCR system and all. So we have discussed about the different G proteins and a basic understanding regarding the regulation of the G proteins. Now in this lecture we will be discussing about the types of the G alpha subunits. Okay. So first we'll write the types of G alpha subunits. Types of G alpha subunits. So first of all, first type will be the G alpha S. First type of the subunit will be the G alpha S, and actually stands for the stimulatory. So S stands for stimulatory. This G alpha S is actually stimulate the adenylyl cyclase enzyme. This enzyme, it is an enzyme which is actually membrane bound. This enzyme is actually membrane bound. So what happens? So if there is a particular ligand receptor interaction, then what will happen? The G alpha S, it will be in their GTP bound form. It will leave the G beta and gamma, and this G alpha S which is present, it will actually stimulate the adenylyl cyclase. So if this is the membrane, and suppose this is the receptor, and if this is the receptor, and suppose this is the ligand, it now binds with the receptor, and when there is receptor ligand interaction, then here normally the G alpha, G beta and G gamma proteins are present. Normally they are present in the GDP bound form, and after that when there is an interaction, so now what will happen? Then it will bound with the GTP bound form. Alpha protein will bound with the GTP, and it will be in the GTP bound form, and it will leave the beta and gamma. Now this G alpha GTP will actually stimulate. There is a particular membrane bound enzyme. So I will just draw the membrane bound enzyme over here. Like suppose this is the membrane bound enzyme. Okay, and this enzyme is known as the adenylyl cyclase, AC. So this will actually stimulate this adenylyl cyclase. Now when adenylyl cyclase is being stimulated, then what happens? This adenylyl cyclase actually converts ATP to cyclic AMP. Now this will convert ATP to cyclic AMP, and this cyclic AMP, after that it will actually activate the protein kinase A. It will activate the protein kinase A, and now what will happen? Now this protein kinase A which is present, this will actually activate, or it will phosphorylate different transcription factors. Okay, it will also open sodium channels, okay, potassium channels and all, K2 plus channels and all. So these are the downstreaming pathway. That is the very important point. Now what happens is that how this protein kinase A actually activate those things. So protein kinase A, this is a regulatory enzyme. It has two catalytic subunits, and it has two regulatory subunits. So these are the catalytic subunits and these are the regulatory subunits. Now when cyclic AMP binds with the regulatory subunits, these regulatory subunits actually falls off. And as a result of that, only these catalytic subunits are present. Only the catalytic subunits are present, like this, and this is the active form. And now this active form, this catalytic subunit of the protein kinase A, they actually now enter into the nucleus. And what did they do? They actually phosphorylate a particular protein. After entering, this is a kinase, so it will phosphorylate. So it will phosphorylate a protein known as CREB. Full form of CREB is very important. It is cyclic AMP response element binding protein. Binding protein. Now what will happen? This crepe protein will be phosphorylated, and this phosphorylated, and this crepe protein, when which is phosphorylated, so this phosphorylated crepe protein will actually now recruit different co-activators. It will recruit different co-activators, and as a result of that, it will perform this kind of functions like activation of the transcription factor. It will also open sodium channels, calcium 2 plus channels, and all. So this is the basic mechanism how this protein kinase A activates. So it actually, after binding with the when this protein kinase A is activated by the help of the cyclic AMP, the regulatory subunit falls off and only the catalytic subunits are present. This is the activated form, and now what happens? It enters into the nucleus. It phosphorylates the CREB protein, that is the cyclic AMP response element binding protein, and this phosphorylated CREB actually recruits different co-activators and further activates different transcription factors, and result of that, different genes, transcription of different genes takes place. So that is important. Now, another important is the different toxins which actually affect this phenomena. Now first we'll study about the role of cholera toxin. Role of cholera toxin. What happens? Once again, I will just check that's recording or not. Yeah, it's recording. Sorry. Now, role of cholera toxin. It actually ADP ribosylates. It ADP ribosylates the G alpha S. G alpha S. It is ADP ribosylated by the cholera toxin. So, cholera toxin comes from the bacteria Vibrio cholera or Vibrio cholerae, and it actually affects the intestinal epithelial cells. It affects the it actually affects the intestinal epithelial cells. It affects the intestinal epithelial cells, and as a result of that, we know that it actually causes diarrhea. It actually causes diarrhea. Now, when it ADP ribosylates the G alpha S, the G alpha S is locked. When it is ADP ribosylates this G alpha S which is present over here, now this is locked. This becomes locked in its GTP bound form. It is bound in its GTP bound form. So when G alpha S is locked in its GTP bound form, this is now, it is locked in this way. So now this is locked. This one is suppose locked. Okay, this one is suppose locked. So this is the locked. Now that is the most active part. So this G alpha GTP, it is the activated form. So it's locked in its activated form. So there will be, as a result of that, there will be continuous activation of the adenylyl cyclase. So there will be continuous activation of adenylyl cyclase. As a result of that, CMP, cyclic AMP, they will be always high, and always, and if it's high, it will activate the protein kinase A. Sodium channels will remain open. So sodium channels will remain open, and it will lead to diarrhea. It will lead to diarrhea. It will lead to diarrhea. So that is the role of the cholera toxin. Okay, so that is an important point. Now let's come to the next. That is the next type of subunit. Second is G alpha I subunit. Next is the G alpha I subunit. This G alpha I is actually inhibit, in its activated form, it actually inhibits the adenylyl cyclase. So when it's in its activated form, when it's bound with the GTP. So when signal is present, then G alpha I is present in its GTP bound form. G alpha I is present in its GTP bound form, and that is the actually the active state. That is the active state, and when it's active, it actually inhibits adenylyl cyclase. It actually inhibits the adenylyl cyclase. Now the point is if it is inhibited, so if it is actually inhibited, what will happen? So it is now inhibited. So as a result of that, no cyclic AMP synthesis. There won't be any kind of cyclic AMP synthesis, right? If signal is present, when signal is present, when signal is present, then what will happen? Then, sorry, signal is absent. So if there is no signal, so G alpha I will be present its GGP bound form, and this is the inactive form. If it's inactive G alpha I, it won't inhibit. As a result of that, adenylyl cyclase, it will be active, and there will be cyclic AMP synthesis. That is the case of the G alpha inhibitory. So when it's in active form or the GTP bound form, then what happens is that it actually inhibits the adenylyl cyclase. Now there is an important toxin present over here as well. So what is the toxin? Now the toxin here is the pertussis toxin. So we will study about the pertussis toxin. So the role of pertussis toxin. Pertussis toxin. It is a kind of exotoxin. Exotoxins are mainly released from the gram-positive bacteria, whereas the endotoxins are released from the gram-negative bacteria. So it's a kind of exotoxin. Now this pertussis toxin is released from a particular bacteria that is known as the Bordetella pertussis. So Bordetella pertussis, okay, they actually release this pertussis toxin, and this Bordetella pertussis, okay, release this pertussis toxin, and it leads to whooping cough. It leads to what? Whooping cough. Okay, actually what happens is that there is a colonization of these toxins in the respiratory tract, and as a result of that there is a whooping cough. This toxin, it ADP ribosilate, it actually ADP ribosilate, okay, ADP ribosylates G alpha I in its inactive state or in the GDP bound state, in this state. So as a result of that, G alpha I is locked in this form. It is now locked. So it is locked in its inactive state. Inactive state. So as a result of that, adenylyl cyclase is not inhibited. It is active. As a result of that, there is cyclic AMP productions. Okay, and as a result of that, whooping cough actually takes place. These different channels are being opened because this G alpha I in its inactive state, it is being locked by the ADP ribosylation by the help of this pertussis toxin. This was actually important. Okay, now next type of G protein is the G alpha T subunit. It is seen in the rhodopsin case. It is seen in the rhodopsin receptor. Okay, so we will see that. Now what happens is that, suppose we have the membrane. Suppose this is the membrane, and suppose this is the receptor. So I am drawing the receptor over here. Suppose this is the receptor. Obviously there will be a serpentine receptor, so there will be a particular membrane spanning domain. So I am not drawing that. And suppose in presence of light, so suppose there is light. Okay, so I am just drawing this is, suppose this is the light. So in presence of light, what happens? This receptor is actually activated. Okay, and this is the rhodopsin receptor. This is the rhodopsin receptor. Now, here the different G alpha, T, beta, and gamma, they are actually present over here. When they are, see this very carefully, when this light falls, these receptors become active, and as a result of that, G alpha T bounds with, normally it was bound in the GDP bound form. After light, it bounds with the GTP, and it becomes dissociated with the beta gamma. Now, there is a particular important enzyme present in the membrane as well. This enzyme is very important enzyme. The enzyme is phosphodiesterase. This is the phosphodiesterase enzyme. When G alpha T becomes bound with the, actually they bound with the GTP, it becomes active. Now it actually activates or stimulates this phosphodiesterase enzyme. This phosphodiesterase enzyme actually converts cyclic GMP, not AMP, GMP, cyclic GMP to GMP. So there is a decrement. So there is a decrement or decrease in the cyclic GMP level as it is being converted to GMP. It leads to, so this leads to the closure of sodium plus channels. It leads to the closure of the sodium plus channels. Okay, it leads to the closure of what? Sodium plus channels. So that is the response. If there is light, the rhodopsin receptor present over here. So right, so when light falls, then this G alpha T becomes activated, and it stimulates the phosphodiesterase enzyme, and it actually converts the cyclic GMP to GMP. And as the level of the cyclic GMP is lowered down, there is closure of the sodium plus channel, and we can detect the light. So that is the receptor mechanism. So when there is no light, then this G alpha GTP will not be activated, and the cyclic GMP level will be high, and the sodium plus channels will be open. But when there is light, there will be closure of the sodium channels. That is an important point you have to remember. So different, you can see that G alpha S, G alpha I, they will activate and inhibit the adenylyl cyclase. G alpha T, they will actually activate the phosphodiesterase. Now the next kind of G alpha protein. The next type of G protein is G alpha is the G alpha Q subunit. They actually stimulate the phospholipase C beta. This in its active form, it actually stimulate the phospholipase C beta. Okay, and this phospholipase C beta, when it gets activated, it actually breaks the PIP2, phosphatidyl inositol diphosphate. PIP2 means phosphatidyl inositol diphosphate. Okay, diphosphate to IP3 and DAG to inositol triphosphate and DAG. DAG means diacyl glycerol. Okay, so these things are very important. This diacyl glycerol is actually kind of hydrophobic, whereas this phosphatidyl inositol triphosphate is a hydrophilic. Now we'll see that in case of the diagram, we'll see. So let's draw the diagram and you will understand. Suppose this is the membrane. And suppose this is the receptor. I will draw the receptor like over here. Suppose this is the receptor, and Suppose this is the receptor. Okay, so this is the receptor present over here, and suppose we have the ligand. So this is, suppose this is the ligand. Okay, so when there is ligand receptor interaction, what happens? Normally it was present in G alpha, GTP bound form, beta and gamma was present. So now what happened? G alpha, this is Q, it binds with the GTP, it gets activated. Now there is a particular membrane spanning enzyme, that is the phospholipase C gamma. So this is the enzyme. This is what enzyme? Phospholipase C beta, not gamma, sorry, C beta. Now what happens? It actually breaks the PIP2 to diacylglycerol, now which binds with the membrane like over here, and IP3. This DAG, diacylglycerol, they actually now activate the protein kinase C. They activate the protein kinase C, and it actually induces the activation of different transcription. Sorry, it actually induces the transcription of different genes. So it actually induces the transcription of different genes. We don't need to know about all the different genes over here. Now this IP3, which is now broken from the PIP2. Now what is the function of IP3? IP3 will go to the smooth endoplasmic reticulum. It will go to the smooth endoplasmic reticulum, and it will release the calcium 2 plus from the endoplasmic reticulum. So this, suppose this is the endoplasmic reticulum, smooth endoplasmic reticulum. So it will be like this, kind of this. So if I draw the endoplasmic reticulum, 1 second. So suppose this is the endoplasmic reticulum. So this IP3 when goes over here, so when IP3 goes over here, so as a result of that, calcium 2 plus is released in the cytoplasm. Calcium 2 plus is released in the cytoplasm. This is the ACR, and this in the cytoplasm, this is actually the cytoplasm, right? So now in the cytoplasm there are different proteins known as the calmodulin proteins. These proteins are known as the calmodulin proteins. They actually bind with the calcium. Okay, they actually bind with the calcium, and calcium is important for many kind of functions, right? Because different, we have studied in the cell biology that calcium, which will, in the cytoplasm, will be used by the calmodulin. Okay, this will be also used by the calnexin proteins. Okay, calnexin, they'll also used by the calreticulin. Okay, these are the important uh, chaperones that are important for the folding of the proteins, right? Calreticulin, right? So they'll be also taken by the calmodulin proteins, okay, for the storage of calcium in the cytoplasm. Okay, and they will also helps in the calcium 2 plus, also helps in the activation of the acetylcholine. It also helps in the activation of acetylcholine, but activation of, not acetylcholine, I'm really sorry, of ACTH. Okay, adenocorticotropic hormones, right? And it also activates, it helps in the activation of different protein kinases. It also helps in the activation different protein kinases as well. Protein kinases, this is also important. So that was all about the G alpha Q. The main important you have to know, and this is the ligand, and this is the receptor. This is also G protein coupled receptor. So in this way, there are different types of G proteins which are associated over here. So that is the main thing you have to know. This was all regarding the G protein coupled receptor. The different types of G alpha subunits are G alpha S, G alpha I, G alpha T, as well as G alpha Q. These are the basic things you need to know. So in the next lecture, we will be discussing about the receptor tyrosine kinase, which will include different types of receptor tyrosine kinase. Will actually discuss about different pathways, like we'll discuss about the RAS MAP kinase pathway, PI3 kinase or AKT pathway, mTOR pathway, and as well as IP3 DAC pathway. That will be all for your cell signaling as per JAM syllabus and PIFS syllabus. You need not to know many more details. There are 2-3 points I will mention, that some important other important points, but basic things you need to know about this RTK pathway as well as this GPCR. So this GPCR or the G protein coupled receptor is being completed. Now in the next lecture we will be discussing the receptor tyrosine kinase pathway. So have a nice day. So we'll meet in the next lecture. Bye bye.